Okay. Great, you guys ready? Welcome back. Day 2. Okay, thank you. All right, so today we're going to do some basics of RNA-seq. I imagine a lot of you know something about it, but just to kind of establish a rigorous framework of basics. And then we have a file system game, which is a game we made up, that Miriam, my postdoc, made up last year to make sure that everybody really has wrapped their head around how to navigate a Linux file system. I think I mentioned yesterday, like that's been one of the biggest struggles for students with this class. So we've tried to come up with fun and creative ways to make this accessible and to help students learn. So team up with somebody maybe who has a little more Linux experience than you if you don't have any, and we'll be giving out candy. Mid-morning break, get your sugar rush on. From there, we will go through a software overview and talk about all of the software packages we're going to use in this class, and kind of why we chose them and why it matters what you pick. And then finally, we'll actually start processing some data. Finally, yes, set up our directories, do some quality assessment of our data, transfer files around, and trim them. Let's see. A little bigger. Yep, so the slides are on, oh, and yesterday I forgot to give you guys an email homework. So if you attended, you'll get your full credit. Thank you, you emailed me something. You like made us something, email me. But I didn't tell you what to email me, so that's fine. I did put for the rest of the days of the course what to email me if you're an enrolled student to get your credit for showing up and doing the work. Okay, so basics of RNA-seq. I'll talk briefly about Illumina as a platform, which is probably what most people are using. The FASTA format, the FASTQ format, a little bit more about RNA-seq, and then I will describe our data set. So Illumina is probably, not probably, it is definitely the most common platform that people use to do RNA sequencing. It's kind of interesting to look at it from the point of a business person or a finance person. I have thought about investing in Illumina because they have been really gangbusters for about 10 years now. Ever since they entered the market, it has been an amazing takeover of the DNA sequencing and RNA sequencing market share. They made $2.4 billion last year, and they are currently estimated to absorb 90% of the sequencing market, which is pretty impressive considering they only came on the scene in like 2005, 2006. So they went from 0% to 90% in 10 years. Why are they so popular? Low price. Really beat out the competition on pricing. They're also very, very high throughput. When I was a PhD student, we were using Sanger machines. These blow those out of the water. So that was very exciting. They have very high base calling fidelity. So high quality base calling. Every base you get off, you have some reasonable amount of confidence that it's real. And we'll talk about quality values and how to tell if your data is good or not. They also introduced, well, they were probably not the first company to have it, but they have paired-in sequencing. They've really pushed the boundary on the length of the reads you can get for your pairs. I do think it's, they're still growing. So from 2016 to 2017, there was another 5% increase in revenue. I think it's pretty interesting that a significant portion of the market is consumables as opposed to actual instruments. The instruments are quite expensive. Some of them over $1 million, some of them significantly less. But they really make a lot of their money on consumables, which when you have to pay for library prep costs, you will appreciate. Yep, and they're all over the world. So they're making inroads in continents, all different continents. So a very interesting company. And this just kind of further illuminates where we are with the sequencing prices and sequencing throughput. The cost to sequence a human genome, our first human genome was in 2001. It cost over $100 million. We are currently very closely approaching that $1,000 mark. And this is just to generate the data, not necessarily to do your bioinformatics. And you can see this turning point when next generation sequencers came on the market. There was the Roche 454, that was a valid instrument for quite a while. They're currently no longer making them or really supporting them, which has further led to Illumina being the only game in town. Anyway, this is where Roche 454 and Illumina came on the market, and you just see this like precipitous drop off in cost. So that led a lot of people to get excited and do a lot of sequencing. But what this means is that lots and lots of labs can afford sequencing, but informatics is now our primary bottleneck in getting all of that data analyzed and in getting everybody trained in how to analyze it, because this is something that happened so fast in the past 10 years. We have very few master's and PhD programs in bioinformatics in this country, and we have a lot of faculty and staff who are trying to pick this up as a skill as well as students, which is one of the reasons we run this as a summer workshop, shorter format, more accessible to a wider variety of academics, not just students. Illumina does offer a wide variety of different instruments, and even for RNA-seq, your decision on which instrument to use can be an important one. So there's the MySeq, which we have 2 MySeqs on campus, and this would be a much smaller throughput instrument, but you do get the 2x300 base pairs, which I'll talk about what a paired in is now. But essentially for each fragment that goes into the machine, you get 600 base pairs back out. These are prices, I think I put down here, from the UT Austin Genome Sequencing Analysis Center. I find them to be middling in price. They're probably not the best, but they always have updated prices online, so it's really convenient to look it up, throw it into a grant proposal, and you can usually get something that's reasonably in this vicinity. That's going to give you 22 million reads. Moving up to the HiSeq, you're going to get shorter reads, but much, much, much higher throughput. For less than double the cost, you get more than 10 times as much data, so 10 times as many reads. They are shorter, but 240 million reads at this point. NextSeq is one of the newer instruments to come online, the NextSeq 500. Again, somewhat shorter reads, double the price, and you're getting another big increase in the amount of data you get out, so 330 million reads. There's also a very, very new instrument called the NovaSeq. Came out less than a year ago. I don't know anyone who has one, but it's supposed to be the next big high throughput. Like, it'll leapfrog this again, and you'll get even more data. So keep your eyes peeled for the NovaSeq.
How many RNAs are there per cell?
That's a good question. Single cell, so there are single cell transcriptome preparations, and I don't think you would sequence it on a HiSeq. Like, I think you would put it on a MySeq. And if you could do PCR free, which would be hard with the single cell.
Oh, but I mean, just estimating ballpark, is 20 million enough to get a sense of what the expression is from the cell? 20,000 genes. That's it, right?
Yeah. So a lot of, so you're almost never sequencing like one cell. It's a somewhat, you shoot for a homogeneous population, but you don't get it, right? Like if we shove a leaf into, or take a whole leaf and do RNA extraction, we get lots of different things going on different parts of the leaf.
You're going to cover the whole genome as well as get a good range of expressions from a.
Yeah, yeah, and I'll talk about that. There's some debate. I think 10 million reads is about the sweet spot for the statistics. The statistics seem to work out well at 10 million. Are there 10 million transcripts in a cell? I would guess no, but I mean, there's probably a lot. But part of it is sampling your variability, which I'll talk about more, I think, tomorrow. So 10 million, so the ENCODE consortium came out with their recommendation of 10 million if you just want highly expressed genes, 100 to 110 million if you want the really lowly expressed genes. Because some things are like 1 in a million, 1 in 2 million, like they're very lowly expressed. And if you want to look at differential expression of those, you've got to sequence much deeper. So it kind of depends on what you're looking for. Jim?
So when over the situation you want a longer piece that in the over the situation where you want a shorter piece but more top.
Well, it's funny because your projects are poster children for wanting longer reads. Right. So I generate longer reads for a lot of my projects as well. If you don't have a reference genome, and you have to assemble your transcriptome from scratch, it's much easier to assemble it from bigger pieces. If you have small pieces, you can imagine like if somebody gives you a puzzle that's been cut into 100 pieces and the exact same puzzle cut into 1,000 pieces, it's a lot harder to do the 1,000-piece puzzle than the 100-piece puzzle. And that's not a great metaphor, but if you have longer pieces, you can find the overlaps between them much easier. You can find, if you have genes that share common domains, hopefully you've sequenced through the shared part of the genes and gotten some unique bases that can help you differentiate different genes. So mostly you want longer things if you're gonna do your own assemblies. Or if you're working with a polyploid. So if you have a which you do both. So when I said you were the poster child, I meant it. So if you have a polyploid and your genomes haven't differentiated too terribly much, and you can use that link to your advantage, if there's any unique differences between the genomes, you can hopefully use that link to sort out which subgenome your read maps to. So those are the two big use cases for longer reads. If you have something like Wenjing is working on chicken has a great genome, super well annotated. She could probably get away with 50 to 75 base pair reads, no problem. Just because it's well annotated, it's diploid. Most of her reads are gonna map uniquely. So that's the difference in my mind. Like I will usually pay extra and take the throughput hit on the MySeq if I have to do an assembly. Yeah. MySeqs are pretty common. So MySeqs are only like $250,000, maybe even less now. HiSeqs and NextSeqs are more on the order of like 750,000 to $1 million. So you get a lot of universities like us. We've got 2 MySeqs on campus, but we do not have a HiSeq. We used to have a HiSeq at Oak Ridge, but as far as I'm aware, it's not running anymore. So you usually get the universities that have HiSeqs and NextSeqs. The only way to make it a viable service core that's self-supporting is to buy like 10 of these instruments and get 10 people in the lab and just make it like a business. Because the instruments are flaky, they have really, really expensive service contracts. You have to have people come in. You have to expect out of 10, probably 1 to 2 instruments is down at any given time. So if you've got customers queued up, you need to have, from what I understand, I used to work at a sequencing service core at Clemson. The only way to do it is to scale up. So only a handful of universities has really invested. And like, we're gonna go into the sequencing game. We're gonna run a big core. We're gonna make this work. And you will see ones that maybe they have a one-off, like a single HiSeq or a single NextSeq. I'm wary of those because if the instrument's down, you have to maybe wait months for it to be back up and get your sequencing data. And a lot of time they're subsidized by the university, which is great. So they'll have really good internal rates for their faculty and staff. But if you're going in as not an employee of that university, then you may be paying a lot extra. So it's good to shop around, figure out what the prices are. Yeah. So that's when I would do the long reads versus the short reads for sure. Okay, so it's probably a good idea if you haven't to learn a little bit about how the Illumina chemistry works. I'm not going to talk too terribly much about it, except as it kind of impacts downstream analysis, but they have some really awesome videos where they visualize what the DNA looks like and how it attaches to the little flow cell and how it bends over to do the paired end sequencing, and the videos are super fun. So I definitely encourage you to watch them. But to give you the basics of what your fragments look like before they go on the instrument, you get RNA fragments, and it's pretty similar for DNA. Some type of fragment, usually pretty short. Standard protocols might be as short as 150 bases, but you can go up to 500 bases or more. So you want a pretty tight fragment size selection. There's a couple ways to do that. But whatever you pick something kind of maybe 400 to 500 would be a good fragment size. You ligate on a universal adapter. This is going to help the machine to take each fragment and separate it and sequence it. You also ligate on an indexed adapter. And what I mean by indexed is there's a special 6 bases in the middle here that vary for each of your samples. And there's also this really great Illumina TrueSeq DNA adapters demystified. So if you get back funny data and you can't figure out what your adapters are, where they go, that's a great resource. But I want to talk a little bit more about this indexing because that's really essential for what we do downstream. Okay, well, first I'll mention paired in. So for the paired in libraries, essentially what you're going to do is you're going to take your DNA fragment, and the instrument is going to sequence it from this direction, and it's going to flip the fragment over, and then it's going to sequence it from this direction. So you're going to get two fragments back, one going in one direction and other one is reverse complemented because it's sequencing from the other direction. And if you were to envision aligning these back to the reference sequences, you don't know what's in here because you don't necessarily sequence all the way through. So you just have some unknown bases. And when you're mapping that back to your reference sequence, you're gonna have one end that maps and hopefully another end that also maps downstream. And then, some kind of bases in the middle that you don't know what they are. This is super helpful. For example, if you have a repeat, you can imagine like this paired in. If you just had the blue read, you wouldn't know which repeat it originated from. But because you have the orange read, you can anchor it to the correct location. So that's why paired in sequencing is good. And we would love to have longer reads. We just currently, with the Illumina technology, we just can't get reads any longer. So this is kind of their fake way of making things longer, is that you've got these two reads. They're actually, but that you know where they are in relation to each other. So informatically, you can use that positional information when you're mapping the reads back to the genome.
The gray length,
The
gray.
We have no idea.
We don't know. Right?
We don't know. We don't sequence through it, and you can.
Two samples, right? Left and right.
Yep, you'll get forwards and reverses. Yes.
What's the major limitation on extending reads? I've always heard that we can't.
Yeah, I think the fidelity of the polymerases as they build the molecule longer and longer, they fall off, they get corrupt. And you'll see with your reads, they get worse and worse and worse in quality toward the end. And I think it's because you have a glass slide, and as you're building this and you're imaging the base additions, the fluorescence of the base additions from underneath, it just gets too long and you can't image it very well and it gets fuzzy. So the new long read technologies, which are nanopores, completely different approach. Like they've almost completely discarded the Illumina approach, and they have little nanoparticles that ratchet the DNA one base at a time through and measure the electrical discharge as each nucleotide goes through. And so it's a completely different technology. So from what I've heard, this technology will never yield long reads. The industry has started over too. And the problem with the nanopore sequencing, which you guys may have seen PacBio, Oxford Nanopore, you can get super long reads. You can get 50,000 bases, but you're getting 15 to 20% error, which is a lot of error. With Illumina, you're getting under 1% error. So it's trade-off. And so a lot of projects, especially genome assemblies, you're looking at a hybrid approach. You want to do a little bit of both. With RNA, it's pretty common to just stick with Illumina. Because transcripts are already pretty short, the short reads aren't as big a problem like they are for like genome assemblies, which is quite a problem. Yeah, so if you do just a mechanical shear and then run it out on a gel, it should be pretty random. It won't be, it'll be pretty random. We see some sequencing bias toward one end of the gene. I think you get a little bit more sequencing depth at the 3' end, but it kind of washes out in the statistics because it's consistent. At least it's right. At least it's consistent across all your samples. But yeah, so you're not doing like a restriction enzyme or something that would be biased. You are usually doing a mechanical shear, so it's pretty random. Or they have an instrument called the Covaris, which I think does the shearing, can do the really high quality, really tight. And that's one of the reasons, right? Like when you're doing your informatics and you're mapping these back, it's nice to know your fragment length. It's nice to know if these two things should be 50 bases apart, 100 bases apart, or 200 bases apart, because it makes your mapping more accurate. So having that tight fragment is very helpful. Okay, so back to that barcode that I mentioned, because normally most sequencing facilities are going to take care of all of this for you on the back end, and you're never going to see it. But once in a blue moon, you'll get your data back and it hasn't been demultiplexed. So I want to explain how multiplexing works so that you'll know how to handle it. So imagine if you had two samples. Let's say you have an experimental treatment and a control, and you extract your RNA. On this sample, you ligate your index. The black part is the same for every library, but it has this little 6 base pair in the middle encoded with TGA, CCA. Great. With your pink sample, or your experimental or your control, whichever one, it's just different, you ligate an adapter, but it has a different 6 base pairs in the middle. In this example, it's CGA, TGT. You then sequence them as a big pool. You mix these two things together and sequence it as a pool. Because the truth is, if you're running a HiSeq, you don't want 220 million reads from one sample. You probably want 220 million reads split across 10 samples. So this is how you're gonna do it. You're gonna take your 10 samples, give them each different barcodes, and mix them together. Then when you sequence them, you'll have these nifty little barcodes, and you can figure out what read goes to what sample. So sometimes you'll get the data back and it'll just be like one giant file. You're like, oh no, which one goes to what sample? You have to use software, and there's a number of toolkits to do this, to figure out your barcodes, and then you can sort your reads into the different samples. So that's a typical way that you would do it, and that's gonna allow you to split your sequencing in much more creative ways. You can run as many samples as you want on a lane or on your instrument. Which brings up probably the #1 most common question I get. Maybe not #1, but it is up there. People are like, what is a run versus a lane? I don't understand all this terminology. And it depends on the instrument. So the MiSeq does not have lanes. It is one run, one input sample. You can have multiple barcoded libraries in your input sample, but it's just one thing. On the HiSeq, the chips come with these 8 lanes. And so when you go to like UT Austin or one of these places, you're normally ordering by the lane. So you can have 40 RNA-seq samples, and they're gonna mix them together, and they're gonna inject it into just this one little channel for sequencing. So if someone's trying to sell you a whole flow cell, that is 8 times as much data as one lane. So they're actually gonna get 240 million reads from each of these individual channels. Just a ton of data. And I think for the NovaSeq, it's the same. You have lanes. So again, I haven't seen any NovaSeq data, but.
So just a quick question. So for the HiSeq, so each sample goes to one lane, or
no, you can put lots of samples, and you're going to want to normally.
So what's the how many samples you put one lane?
We've gone up to like 20 samples in a lane without a problem. It depends on, you have to, you back calculate how you want to do your, like how many reads do you want per sample?
20 samples, you can get more than 10 million reads, like you said.
Yeah. So this is one of those things where you want to work with a really high quality sequencing facility, because I got some data back from, maybe I shouldn't say names, I shouldn't be throwing other universities under the bus, but I've gotten data back where they mixed my 20 samples, and some samples had almost no coverage, and 2 or 3 samples ate up the whole lane. And it was really frustrating. But UT Austin, which I've used quite often, and a number of other places, they do a really nice job quantifying the libraries to see how many fragments they are, and then balancing the mixture, so that you get extremely consistent number of reads per sample. But you have to be working with a facility that's good at what they do. And most sequencing facilities are fine. Only once have I been burned and gotten back reads. And occasionally you'll still have a library failure. Like sometimes a library just goes kaput and you don't get anything. But for the most part, you can get within 20% variation of the number of reads per sample. So a little bit of variation, but not a ton. Yeah, and I know you can. I mean, the Amplicon 16S people, they'll multiplex 200 libraries on a MiSeq. So you can go up to like really big numbers. I don't see that as often with RNA-seq, because normally you want the coverage as well. But
yeah, for RNA-seq, probably you want more reads.
Yeah. So in particular, 16S comes to mind because you can publish papers with each sample having 10 to 15,000 reads, which is not going to happen in the RNA-seq world. So yeah, 5 to 10 million per RNA-seq is currently typical. Although, you know, we published papers when I was in grad school with like 10,000 RNA sequences and thought we were high throughput. So things do change quickly. You have to keep an eye on that.
That 8 lanes is essentially 30 million that you get with MySeq. It's like 8 MySeqs.
Yeah, oh gosh, it's like 10 times 8, so like 80 MySeqs on one run. And some of the HiSeqs have two flow cells, so they're actually running 16 lanes at a time. So it would be 160 times as much output as a MySeq. Yeah. But I think the HiSeqs take like 11 to 14 days to run, and the MySeqs I think are a little faster. So
11 days to run.
Yeah, they take a long time. Yeah. Some of the new ones are faster, but the HiSeq 4000s, if you do a paired in 2x150, it's like 11 days to run. It's a whole process, and you have to wait. But they're getting so much data. I mean, they're getting billions and billions of reads out of those 11 days, so they're pretty happy with the output.
Yeah. So just kind of thinking through the last 4 slides, YX and treatment experiment, right? Send all the treatments down there. So they wrap them, index number or index beats is added associated with each treatment.
Yes.
So they put everything together.
Yes. Okay. They mush it together into one tube and then like pipette it straight into this lane. And usually this is Jim's lane, and this is Vitaly's lane, and this is Meg's lane. Like a lot of times you're sharing that with a bunch of other people because it's rare. I mean, I guess if you work certain places, maybe it's not rare, but for us, I would think it's rare for anybody to buy 8 full lanes. It's just so much data. Yeah. Yes. So the RNA-seq library preps, for those of you who haven't ordered your RNA yet, these prices that I gave you earlier, the $2,000 to $6,000, that's just to run the instrument. It's like $200 to $300 to actually do your library prep as well for each sample. So it gets really expensive really fast. Right, so when you come talk to your resident bioinformatician, and she's like, you need like 50 samples. And then you're like, oh no, I can't afford that. This is the ongoing debate. And I think I talk a whole bunch more about this tomorrow. I mean, but we can talk about it now. It's just library prep is still a huge bottleneck. So there are people, so I work with Jason Holliday at Virginia Tech. And he sent me this paper, and I can't remember the first author, but they've published this incredibly cheap RNA-seq library prep. It's like $20 a sample. If your labor's free, right? Like labor's never free. But in this paper, and so he tried it, and he sent me 10 libraries, and they were complete crap. Like they were awful. The reads didn't map to the genome. They were like low quality. But it gives me hope that if somebody sits down and optimizes that, we can get cheaper RNA library preps. But right now, as far as I can tell, it's still worth it to pay for the real deal. Because
the library prep is from the extract total RNA, the sample ready.
No, that usually doesn't include extractions. Okay. You send a tube of RNA. They'll do
Is that the mRNA or
total RNA? Okay. I usually pay for the sequencing facility to either do an mRNA selection or a ribo removal. But you can do that, so it kind of varies, like what you want to do in your lab versus what you want them to do. There's a price point for each step. The more you want them to do, the more you pay. And that's another reason that the UT Austin wiki with all their prices is really nice, because they break down, like, we'll do your sample QC, we'll do your ribo removal or your mRNA selection, we'll do your library, so you can kind of see what each piece costs and make decisions based on that. Any other questions? Okay, so I want to talk a little bit about file formats, which are boring but are really important for bioinformatics. Okay, so first, the FASTA format. The FASTA format has been around since the 80s. It's been around since sequencing was invented. And it's super simple. It's basically, you start with a line that begins with a greater than symbol. And that's called your header line or your comment line. And that has some sort of unique sequence ID. This is an example of something that you would download from NCBI. They have quite a complicated header line with lots of information. But you can also see something as simple as like read one. It doesn't have to be this long thing with bars, and NCBI tries to shove in like the accession number and it puts a description after. So the header line, you have to have your unique ID with no spaces, and then the rest of the line you can put whatever you want. So if you need to add comments or you want to add information about what the sequence is, you can use the rest of that line to do that. Following is the sequence. And so this is valid for amino acids or for nucleotides. You'll see FASTA files for both. And the sequence does not have to be on one line. It can wrap over multiple lines. You will know that you're starting on a new sequence when you see a new greater than. So the greater than will always be at the beginning of the line, and it will always be the first thing you see in starting like a new sequence block. So this whole thing over here could be, for example, a sequence file, a FASTA formatted sequence file. There's a couple of different paradigms for what types of sequences. So of course your amino acids are pretty well defined and what you can use. Sometimes you'll see stars instead of that stand for like a stop codon. Some software will take that, some software will not. Sometimes you have to remove the stars. For the nucleotides, there's IUPAC codes. So normally they're just A, C, T's, and G's. You'll also occasionally see N's, and N would be, I have no idea what this base is. There's also a set of bases like K's that stand for A or G. Like, I'm pretty sure it's an A or a G, but I don't know which one, so it's a K. We're gonna put a K in there. So sometimes you'll see weird letters in your nucleotides, and if that happens, you just go look up IUPAC codes, and it tells you what they mean. That's pretty rare. I don't think you're gonna see that, but I thought I'd throw it out there because it happens to me sometimes, and then the software will choke and be like, what is this nonsense? And so what I do is I just change all of the K's into N's or all of the W's into N's. And I forgot to make that a slide, so we can just Google it real quick. Yeah, so this is it. So 99% of the time you're gonna see A, C, G's, and T's. Occasionally you'll get something like this. R is an A or G. Y means C or T. B means C or G or T. So most people are going to use A, C, G's, and T's and N's. And an N just meaning like, I don't know, unknown. So that, hopefully you won't have to worry about this too much, but if you do see something and you're confused, that's probably what it is.
How do you get gaps in the sequence? Can you get gaps? It should be in the sequence.
Can you get gaps? You can. Yeah, so it's pretty rare to see that in Illumina. Sometimes, I'm trying to think, I think you can have instrument problems where like so the way it works is you have a glass slide and each time a base is added at fluorescence and there's a super, super high resolution camera taking pictures. And sometimes if in a region, if you have a low complexity library and let's say everything like 90% of the slide is a G, like everything that's not a G will get washed out in the image and you'll get some N's in the middle. I don't know, it's an instrument error. And a lot of times to avoid that, they'll ask you to spike in X or spike in something to create more nucleotide diversity. So you're not sequencing, you don't want to sequence this. This is problematic for 16S. If you're sequencing almost the exact same sequence every part of the slide, you get weird things like ends in the middle of your sequence, but it should be rare. I don't see them very often. If it's a PacBio or an Ion or something like that, it's much more common, but that's because the nanopores are flaky and honestly, I don't know enough about nanopores to say why, but you're not gonna see that very often. You're more likely to see it if you do an assembly and the assembler has no idea what goes in the middle. It's just like, I'm gonna stick a bunch of N's in there and that's all I know. So, which you would normally see FASTAs after assembly, or after you have, which your raw data that comes off the instrument comes in a FASTQ format. And FASTQ format at first glance looks a little intimidating, but it's not. We can break it down. Every sequence is in a 4-line block. So you'll have 4 lines that correspond to one sequence, and the next 4 lines correspond to the next sequence, and the next 4 lines to the next sequence. You can also tell because the very first line will start with an @ symbol. And this @ symbol basically says this is a new sequence block. This is the name of the sequence. This is a very typical Illumina. All of your Illumina data is going to have this stuff. This is actually a unique machine ID number. So if you go download Illumina data from somewhere else, you can actually track it back. If you know the machine numbers, you can figure out what sequencing facility sequenced it based on this machine number. And this stuff over here is information about, on that little glass slide, where the individual read was located. And you can use that information downstream for some QC. But none of this is really going to matter to you, but that's what it is, so it's not confusing. And then I'll give you a link. The second, so sequence identifier, optional description on the first line. The second line is your actual sequence. Very straightforward, just your nucleotide bases. Mm-hmm. Yeah, they can be up to 150, but they'll be on one line.
Yeah, but 41 is kind of short, so that's weird. Or
so when Illumina's first came out, the maximum read length they could do was 25. So if you go get old data, well, and some people still sequence relatively short libraries. Like I know people who do RNA-seq at 50 or 75 bases. 41 is particularly short. I don't think anyone sells 41 anymore. But I do see 50s still. But yeah, when they first came out, I was like, what are we going to do with 25 bases? It's terrible. So in the scheme of things, they've actually added a lot of length over the past 10 years. Okay, the third line is, it's the stupidest thing. It doesn't do anything. It has to start with a plus, and then sometimes it's a repeat of your comment of your first line, and sometimes it's just a plus and nothing else. This drives bioinformaticians crazy because it's eating up a whole bunch of file storage on our systems that we don't need or want to eat up. Like, there was no reason for this line. But it's there, and it's making our FASTA files bigger than they should be, our FASTQ files bigger than they should be. And I don't know who made this up, but they should be yelled at. So useless third line. And then the fourth line is this one that looks like complete and total gobbledygook. And what this is actually each individual character corresponds to a nucleotide base, and it encodes a quality value. So it's basically a super shorthand way, instead of having like 10, 20, 21, 15, so instead of having a number for each of your nucleotides, we just use a letter that represents a number. So this is from lowest to highest. So the exclamation point is a 0, so that would be a base that you would definitely not trust, all the way to, like, I like to open FASTA files and I think about, do I see a lot of capital letters? If I see letters, we're in good shape, because this is 41, 40, 39, 38, 37. So all of those are very high quality scores. So there's a logarithmic formula you can use to calculate this. It's a basic Fred score. It takes a little bit of math to get back to it. But you can just remember an I is 99.999% likely to be correct. An A is only 90% likely to be correct. So anything from A to I is pretty good. Anything below that, I'm going to be a little suspicious of, right? Like if it's only like 50% right, just call it an N and move on. But basically the instrument is going to attempt to tell you how confident it feels that it called the base correctly. That's an algorithm that's native to the Illumina instruments, although other instruments will also give you quality values.
How good is that score?
Pretty good. I think they're pretty accurate. They're not perfect, but they're pretty accurate. Oh, well, they take genomes that they know that they've sequenced to 1,000x depth, and they have a really, really, really good idea of what the actual reality, biological reality is. And then they go back and they look at miscalled bases. which you can totally see. So when we align reads to a genome in this class, you'll see like a random base here or there that doesn't line up. You'll have like 40x coverage and one base and one read won't be right. And you're like, okay, that's an error. And then you can go back and map those instances to these numbers and see how good they did. And it's pretty good. It's not perfect, but it's pretty good.
So you said that FastQ comes off the machine.
Yes.
How do you go from Q to A?
Software, lots of software. We'll do that in this class. And Miriam will talk about this after the file system game. But yeah, it also depends on if you want to go to FastA. So if you're not doing an assembly, you don't necessarily care about ever arriving at FastA. All you care about is mapping your reads and getting your quantities out and then doing your differential expression on those numbers. So your end game might actually be to get numbers, quantities of gene expression in each sample. So you may never see a FastA, which is okay too. All right, a little bit more about RNA. So to target our mRNA, probably the most complete way is poly A enrichment. This is going to give you the greatest majority of your sample actually being from mRNA, which makes sense because your processed mRNAs have this poly A tail, and using that as a selection is a very strong way to make sure this is all your sequencing. However, I know some of you said you're microbial. In that case, you will not have poly A tails, and this will not be available to you. So the other thing you can do is ribosomal reduction. So removing the ribosome with rRNA out. If you don't do this, your samples are going to be 60 to 80% ribosomal RNA, which is in general not that useful. So those are the two major ways to make your libraries make sure you're getting RNA. Hopefully you guys are familiar with RNA. If you later on, if you want to brush up on how RNAs are processed and why all of that, why a poly A tail exists, we can go through that. So experimental goals for RNA-seq. Some people just want to do a catalog of genes. Like what genes do I have in this organism? More often you want to see actual quantities of expression. So our gene expression levels, maybe between a control and a treatment. All of the above. The nice thing about RNA sequencing over microarrays is we can discover differences between alleles and allele expression. We can discover the existence and differences in expression for splice variants as well. And neither of those is really feasible with a microarray. We also do RNA sequencing in order to annotate the genes in a reference genome. If we don't know where the genes are, you can use RNA to figure it out. You can also use RNA to do genetic marker discovery, whether you're looking for microsatellites, SSRs, or SNPs, some type of variant that you can use in a pedigreed mapping population or for an association study. And also post-transcriptional modifications and RNA editing you can figure out from RNA sequencing. In this course, we'll pretty much only be looking at differential gene expression levels. But if you have questions about software for any of these other things, just ask me or one of the TAs and we can help you figure that out. Okay, so this is an important limitation that a lot of people don't really understand about RNA. You're not getting absolute quantities, you're getting relative quantities. And I feel like all stats courses start with like you have a bag of red and black balls, you're drawing balls out of a bag. And so we're going to extend that analogy. Let's say I had a huge bin here of different colored balls corresponding to genes. And I have another bin, and it's only half full of different colored balls, each corresponding to different genes. If I sample 100 balls from bin A and 100 balls from bin B, I can give you information about the relative proportion of the colors. What I cannot tell you is which bin is more full. I cannot tell you absolute quantities. And because we are sampling from a larger pool of RNA, and you cannot tell much about absolute quantities of genes. So it's very hard to think about genes like this. There's more of this than of this. You can say there's more of gene A in sample 1 than in sample 2. You can't say there's more of gene A than gene B, because it's just hard to do absolute quantities. Does that make sense from a statistical? It's a sampling problem. So it's important you realize that before you go in case what you really want is absolute quantities, in which case I don't know how you're going to get that. You're going to do RT-PCR or something and have some controls. But RNA-seq is not going to be great for that.
can't you spike give a known numbers?
Yeah, the spike-ins are, yeah, you can. The statistics are hard and I don't think they're that accurate, but that's just my opinion. I think people do a lot of spike-ins in RNA-seq and rarely do they report on them. And the ones I have seen reported on, yeah, I think they're a questionable statistical utility, because I think the spike-ins, like, you're not really sure. You hope you know how much you spiked in, and you hope you know. You also don't like it depends if you PCR amplified your library, and I don't know. So most of the questions that we ask for differential gene expression are, is gene A going up or down in relative expression between my control and my treatment? We rarely ask, is there more of gene A in one sample than gene B? Because it's hard to say if those quantities have changed. There's also reverse transcription, PCR, and fragmentation steps, all of which can introduce bias, PCR probably being the worst. There's depletion of reads at both the 5' and the 3' end. So you will, if you have a transcript, often sequence more in the middle. I actually don't 100% understand why that is, but it seems to be a pattern, which can make it really difficult to identify the true start and end. And you may underestimate the expression level for short genes if if you get better sequencing in the middle and there isn't much middle, you can have a problem. There's also GC bias. Sequencers do not sequence well at very, very high GC or at very, very low GC. If you're working on some extremophile or some organism that has strange GC, that's something to think about. But there are PCR-free preps available. And if you have a plethora of RNA to work with and you're not concerned about like single cells or anything like that, considering a PCR-free prep can really help with a lot of these issues.
I was going to ask on the previous slide, how do you feel about experiments to use housekeeping genes to try to normalize expression levels, or are they using it to try to come up with an absolute quantity? Or?
Yeah, I think you're still using housekeeping genes as, housekeeping genes help. I mean, I do think they do. And I think the gold standard is still to go back to real-time PCR with your housekeeping genes. That's what I see in the literature the most often. I haven't really seen people use housekeeping genes in RNA-seq itself to do relative quantities. Yeah. I think, and I still have reviewers ask me for RT-PCR all the time. So yeah, they really want to see that. So they believe the RNA-seq is real, but they'd like you to back it up with like an RT-PCR with housekeeping genes, which is a problem because my lab we don't have a wet lab. So I'm like, your RT-PCR is not getting done because I don't own RT-PCR equipment. Fortunately, I have a lot of good collaborators who I can call in to help. No, that's something you do downstream. You save your RNA and quantify it later on. I'm hoping, so in my argument, and I have gotten away with this for the past 3 years, if you have enough biological reps, you've done due diligence. You've quantified the variability in your sample. And I think most reviewers are starting to buy that pretty well. So it's more, the PCRs is more like if you have one sample, one treatment and one control, and they're like, right, like, that's statistically going to be problematic. Please go do your RT-PCR and preferably add some biological replicates. So the data set that we're going to be working on today is from apricot trees, and it is from a grant with Bert Abbott at Kentucky is the lead. And the grant is entitled Abiotic Stress Response and Adaptive Phenology in Fruit Trees. But basically we're looking at dormancy. We're really interested in how trees go into dormancy and maintain dormancy through the winter and wake back up in the spring. And this is super relevant for the Southeast right now because we just had a winter with very low chill hours. It did not get very cold. Trees, if it does not get very cold, are very confused. So our varieties of peaches may expect an hour of 400 hours of very, very cold weather. And they're sitting there and they're ticking off. Okay, that's 1 hour, that's 2 hours, that's 100 hours. And at 400 hours, they say, okay, winter's over. As soon as it gets warm outside, I'm gonna wake up. Well, if they never get 400 hours, when spring comes, they don't wake up and they don't really know what to do. And so you will get some blooms, but not very good flowering, not very good fruit set. So we're going to have very low peach and blueberry crops this year in South Carolina, Georgia, and Tennessee because of our super, super warm winter. So this is one of the reasons that we're really interested in looking at this. So it's a much larger experiment with many dimensions, but we've chosen kind of a small subset to work on in this class. We're going to take at 800 chill hours for buds from a 100 chill hour variety, so a variety that you would normally grow in Georgia, and from a very, very high chill variety, so a variety you might be able to grow as far north as New York. So they're calculating dormancy chill hours very, very differently. At 800 chill hours, how are the buds behaving transcriptionally? Even though they're kind of like closed in these little bud scales and they look like they're not doing anything, there is actually some metabolic activity going on in there as they're calculating chill and kind of keeping track of when they should wake up. And so that's what we're sequencing. And this is Jiali's first PhD project. So I'm sure she'll be excited to get all this help on her project. Yeah, so that's the, and we have sampled the data down to like 250,000 reads. So it'll run really fast on Beacon. But we actually have tens of millions of reads across the different libraries. So this is a small subset, but should be interesting to work on. All right, so you guys asked a lot of questions throughout, which is great. Do you have any additional questions? Okay, and we'll get more into experimental design, don't worry. All right. So next, I'm gonna get Miriam to come up here. We're gonna do the file system game with lots of candy as prizes. And yeah, if anybody needs help logging in or still having trouble, you can skip the file system game and come to the back and we'll help you log in.
Okay. For this, I have brought some paper and some pen for those that don't need, because maybe you want to draw the tree or also the command. So if anybody needs some paper or something, just pass them around. Thank you. No sé cómo es. Okay, so I hope you are here. So I have designed only a few exercises for you to get more familiar with the Linux system, mostly for navigating the file system. You can do it on your own, and also you can team up with some others to help you and be more collaborative. And yeah, whenever you're ready. So, yeah. So the first part is about understanding the file system trees, because in Linux, remember, we have a root, and then we're going to navigate from file to file with the commands, knowing where you are with pwd, and also doing ls to know the files that are inside, and using cd. Also generating some files with nano. This is an example of how this first game works. So I will be giving one tree and giving one command, and you have to draw the final tree. Got it? In this case, on orange, you have your working directory, so it is a relative path, and I'm generating this file using the nano command. So this is your first challenge. You're given that tree, and if you would type those two commands, what would you have?
Did the first example make sense?
Yeah.
Yeah. Okay.
So
When you're ready, just put up your green sticky around.
yeah.
To go into the student green fund.
Should we put a timer?
I'm trying. I'll do how it. Oh, there's time up there. 56. Maybe I should be ready in a couple of minutes, more or less.
You think 2 minutes? 3
Yeah.
minutes? So that by 10?
So.
By 10?
By 10.
Be 4 minutes.
Oh, okay. Well, this is the first.
Oh, it should be
the
easy.
second.
Yeah.
It's on the detail.
Yeah.
No, this is for your. Yeah, this is only for drawing.
Yeah, the point is, like, I know that the way I navigate a file system is I keep in my mind where I am in the structure. So this is trying to help you, especially for students who aren't used to navigating this way, think about how to move around in the file system just mentally, not even on the computer.
Well, also, if you were into the computer, it should work. But
Yeah,
it is for you to have a better idea of how the file system looks like. No, you only have to draw it on the paper.
When you say draw, if you do LS, right? Is that what you're saying?
No, I give you a paper, a green sheet. So with pens, you only draw how the final
We probably should have
would
given
look
you
like.
this image. Like,
So
like
it is not
this.
on the computer.
If you made a directory, where would you put it on? If you were in this directory at the command line, and you made a directory, where would you draw it on here? And then if you did nano test and created a file, where would you draw that file on this image? Yeah.
So whenever you have it, put your green sticky, and they will pass around with candy. Oh.
No, like draw the picture and then add where the So if you guys want to grab a couple of candy and yeah, you can use it here. So can you
Yeah, she has a green sticky, and she did it correctly.
Oh,
should I check?
I did it for her. Yeah.
This is what you can do.
Yeah.
Yes. Right. So make your software with you small you're right here.
In this example, you have to draw the result of using this command. Try to again on this.
So she gave you this, and you through this. Now she gave you this, and yes. So you got her tests. So now I directory.
So on the if you try to this command,
And then if you made the file, where would the file be? It's to make you think about directory.
Yes.
and then inside that folder you will generate this file.
easier for people who don't like chocolate. I mean, I don't really support those people.
These are the commands. So,
No.
I will do now.
I have that problem too. That's funny. I wanted that. Like, I like 2 years ago. I went to
Okay, so I will continue forward. This was an example. If you went down, you had the answers. So this is what happened after typing those commands. You should have generated one folder inside the stud folder and then generated one file inside that new folder. So if you have understood now, with the same tree, what will happen when you type these 3 commands?
The orange indicates where you are.
So does everybody understand how it works? Yeah, because what?
I don't know how to
Change.
If you have any questions about the commands, what they should do, feel free to ask.
You should make a symbolic link
No.
instead of copy.
Well, maybe you could have made a symbolic link. Oh, sure, but that is like.
Imagine if ILM goes FA is 10 gigs.
Yeah, of course. In that
And
case,
then you copy the file again.
I usually create symbolic links, but it is for you right now. For example, if you're going to use some of our data, you're going to copy that data. You're not creating symbolic links, probably.
Just once. And just once, not twice.
Yeah.
It's twice. It's 2 copies.
Oh.
One is an test, one is a test.
Yeah. Well, whatever. Are you ready?
I think so.
Where did it go? So how many of you got that? Or aren't still there? Okay, so I'm moving forward. So this is what happened. You're creating a new directory and copying the file. Yeah, you could create symbolic links, so not waste too much storage, but here we're learning the basic commands. And using move on a same file, what it does is renaming that file. So, you do it well? Great. So on the next exercises, it'll be different because I will give you 2 trees and you will have to write the commands. Ooh. First, we're going to work with absolute path names. The absolute path names are those that always start with a slash at the beginning. So you will have to be going folder by folder until you get to the final file. So this is the first example. We're going to remove the red file. So we have to remove the / for the root folder, your first child folder, and then the file. So here is, if you all got this, I'm going to the challenge. So this is the first example. You see what is different between the two trees? What did I do? Great, so how would you write it?
So what's the command to create? Yeah, Victor. Okay, good job. Slash. Yeah. So
Yeah.
did you write it down? Well, no. So this is, you have to do. This is a full path. Write the full path. So how would you, if you were sitting at the gray box, if you're sitting in the root directory, how would you create a folder all the way down inside OO? Student OO. Yeah.
Yay! So, ready for the answer?
Yeah, so if you were sitting here, how would you write the full path?
Okay. So this is the first answer. So you're generating a directory, so make dir / the student folder and the name of the new directory. So if you have this, this is the complicated exercise.
If you don't, you should have a red stick.
If you don't, you could have a red sticky.
You're like,
Are there any, well, if you have any questions, also you can raise your hand. So,
With the red stick.
or put the red sticky. So in here, what happened from one tree to the other tree?
Can you tell them how many commands that would take?
Yeah, you see?
These three.
Yeah, there are 3 commands that you can use to do this.
We don't know if it's the same file. It's the same file, but it doesn't have to be the same.
Yeah, this is the
You
same
can create
file.
it or you can copy. We won't judge. We won't judge you.
Yeah, I kept the name so that they could be easy
We don't know
to
what
see.
goes inside.
Yeah, what's inside? But yeah, they are the same file and the same folder. At least the same folder name.
Those two were copy. It's from where? From where? Where it go?
Oh, wait, should we copy that?
No, you just copy.
No, since he's tough.
You could.
Nope, if you're copying, so you'll have
You could.
command
Okay, but
move something and then be. Yeah, if you wanted to move from one location to another. Yeah.
And then you copy to stuff.
And then we need to create these files.
Yeah, so you created that one, you just need to create this one. So you could copy it, or you just create it.
Also, just for a hint, it is not the same. There is some trick when you want to copy a folder. You have to add something. It's not the same as when you want to copy a file. If you remember.
You don't have to, you already created folders, you don't have to worry about that. Yeah, because you created it in, you created it. So all you do is just create that one. Oh, okay.
But we could do instead of.
Well, you can make this pick, so you can copy it.
We can.
So to copy it, you can copy //file.
order before.
And then you would say you were a copy.
You need to start from above top. Because we're here all the time using absolute value, we have to start from
It'll still be
So
there, yes.
basically,
Move means that it goes away. But then you have
Yeah, but this was
right, to create
yeah, the first one was yeah, the second add this.
So yeah.
Well,
You don't
not
need.
the whole-
Also, do you maybe brain power?
So you copy everything inside.
You want more honey?
So whatever it has, I'll do that right inside.
No, we did.
It's not very bad.
Yeah.
That's all I have to say.
I bet can you bring some honey for honey?
Yes. You want Starburst or chocolate?
People say, that's not
Okay, so how many green stickies or red stickies did you have this?
No,
Yeah, they're not using them. Please use your red and green stickies, so we know how many of you did this. We can move forward. Ring, ring, ring. Yeah, so most of you got it. What?
maybe.
Yeah, what? Yeah, this is. Yeah, but the copy is. This is. No, this is a more. Yeah. Well, I'm going to give the commands, but almost. So these are the commands. You have two different ways. You can either copy the tutorial folder, but you have to remember to use the recursive option. Or you can create the tutorial folder and then copy just the file. And the other step was moving the GFF file inside the hat folder. And that was with absolute path names, but many times we will use relative path names. They are usually shorter and easier. And the relative path names will indicate that you are on your working directory. In this case, you are here. And your commands are going to assume that you're on that folder. So from this folder, this is only foo, this is foo, this is dot dot. So you can go up and down the tree. In this case, if I'm doing this, moving this foo into this other folder, I type like this. I don't need to type the whole path. So here is your first example to start playing with it. Yeah, so what happened? Mm-hmm. You remove the folder.
With everything.
There is only one command here.
A folder with stuff in it. How do you remove a folder?
Yeah.
R-O-R.
Right, because here,
So it'd be.
because now, Slash is kind of.
With this R. Yeah,
Or
yes.
Just R by itself would be something different.
No, it is only an option. It is not a command.
So all the way back.
Yeah.
Or I could be dash RM
No, you have to first the command, then the option, and the argument. Yeah. Do you want to tell me?
What?
Do you want to tell
No.
me? Okay, so this was an easy one, was it? Only remove. It is a folder, so you have to use the recursive option, the full. So that's it. So we're going to do it a little bit harder because you have to copy that Linux tutorial folder inside the working directory. So you have to move up the tree using the dot and slash for each level. And then generate the new file. So how do you do that?
We are in the study.
Yep.
Yes,
Right.
you are.
So if you said,
But there, we just created.
yeah, so we recursively copy this. So we want to copy
One, two,
dot. How many dots?
Yeah.
one, two.
Also, again, you have the option of creating the folder and only copying the file.
Yeah.
It works the
You
same.
hit your tab and you're like, oh, that's not something you just and you're back another one. Yeah. All right. Everyone feel like they got it? Anybody want more help? Anybody need a quick break?
Have it? No.
Okay.
What is the command?
But it's not gonna work.
Tutorial?
You can,
Yeah.
yes.
What is missing?
Go ahead and say this.
Oh, the negative R.
No, yes.
The best thing is instead of thinking about starting by me. That's what I learned.
So then that is going to be in.
No, but this is relative file. So it is only in relation of your working directory.
Create a file which is now.
So, well,
That's the test.
Well, you are here. So you're already here. So this is your PW. So you only have
right, it's all part of this.
No.
no, only this. Because you're
Ah,
already
because
here.
I'm already.
So we're really behind. What do you want to do?
Well, this is, I can skip the last one.
Okay.
We can give them the last one as an exercise, or is it too much?
I mean, you can tell them they can do it on their own if they want.
Yeah, well, I would tell them.
How long do you think the lecture will take?
Yesterday it took me 30 minutes, but maybe I can do it faster.
Yeah, mine took a lot longer this way.
Yeah.
Sorry.
Okay, so I'm moving forward to the answers. And here it is. With only 2 commands, you can copy recursively the Linux folder into your working directory. So yes, you have 1, 2, and 3 levels of dots to go up 1, 2, and 3 folders to the root and then change to the other branch. And then copy Linux tutorial, and the dot means your working directory, which is in this case the student folder. So it is generated here. And then you only have to generate the new file within this folder. The other option was you can create this Linux tutorial folder and then only copy the file without the recursive option.
You have to use one of the text editors to create a file. In both one of the.
Well, you can create it in we have said Nano, but also you can have Couch. only generates an empty file. Also, there is Beam or Emacs. There are other editors. But
Okay,
yeah, it's only that we have said Nano yesterday. So we're not going through this, but if you're interested, there is this final challenge. You have it on. Yes?
Is it grand just for the whole band?
Yeah, I think there might be 5. Yeah, there is 1 command per star. So I think there are 5 commands if you want to do it on your own, but we need to move forward to the next lesson. So, yeah. Did you like it? Do you understand now better how it works? Okay, so. Okay, so here I'm going to explain how the RNA-seq analysis looks like. Usual RNA-seq pipeline looks like this. You have a first quality control step of the reads that come out of the sequencer. We're going to use FastQC, this software. Then you have to trim the adapters that Meg was saying before going forward. You can also do some other quality trimming steps that I'm going to say later. And after trimming, you usually do a second round of quality control to compare how your reads looked before and look like now. And MultiQC is nice software because it combines multiple results into one single file. It is much easier to see. At the mapping step, there are multiple options. We have selected star. It's good software, it goes fast. And once you have your reads aligned, there is some software for visualization. So you get to see how your reads are aligned to the reference. The genes, the reads within the H gene have to be counted for comparing the differences in expression. And we're going to do that with HD-seq. Finally, the differential expression is within R. There are multiple packages in Bioconductor that you can choose, but we have chosen the seq2. Some other steps that can go downstream of this pipeline, but we're not going to do that, is a pathway or function enrichment. Probably you will know about KEGG pathways or gene ontology. And finally, if you're going to publish your results, you need to upload your reads to NCBI. On the general pipeline, we have assumed that we have a reference genome, but many times, mostly if you're working with no-model organisms, you won't have a reference. So another workaround that you can use is to generate your own reference transcriptome. There are also multiple software options, and we are going to use Trinity. So you create a de novo assembly where you can map your reads. To know the functions of these assemblies, you have to perform some annotations. We are going to see BLAST to annotate some transcripts. It is annotation by homology. If you are more interested, there is a nice review on what is a good pipeline to generate a transcript of annotation. So in this course, we're going to cover all these steps on our different labs. So lab 1 will be today, later, for quality control and trimming of the reads. The lab 2 is for visualization, mapping visualization, and counting the reads within the genes. Then we will generate a small de novo assembly, later perform some functional annotation, and finally we're going to perform differential expression. There are many things that you can control at every step. This is also only a small review of what an RNA-seq pipeline looks like. But if you want to see best practices in RNA-seq and also how large is, how many steps you will need to make some decisions, here you have also a nice paper. So on our first step, or quality control, we have the reads that come out of our sequencer. So we want to know first how good did the machine work, and also, like you already know, their quality. The machine. There is a confidence for each of the bases, so you can know overall the reads, the average quality. And also, if you have parent data, usually you won't get the same quality on the first and the second read pair. So from FastQC, you get something like this. You get a threshold, so everything that goes above the threshold indicates that it is of good quality, and below the threshold, lower quality. And from the beginning to the end of the reads. Usually the forward reads have much higher quality than the reverse reads. And also the first part of the sequence gets higher quality. As long as the sequence counts, usually there can be some mistakes. So that's why usually the quality decreases at the end of the reads.
So is this a graphical presentation of that fourth line as the Q plot?
Yes. So this is the average of this fourth line.
And that's 41 nucleotides, right? It's just to make sure there's 41.
Well, in this case,
40,
yeah, it's there are 40.
40 nucleotides.
Yeah, usually when the reads come out of the sequencer, they all have the same length.
When you set a cutoff in green, what's the cutoff? Is it the green cutoff?
Yeah, this is the cutoff. And on FastQC, usually there is like a fixed cutoff. It is mostly for visualization. But where you're going to set it actually is when you're going to trim. Trimming is mostly used for removing the initial and final adapter sequences. So for example, if you are sequencing from Illumina, you can download a sequence file with all those sequences and aligning them to the beginning or the end of your reads. You can remove those sequences. But also, if you want, you can remove some biases based on quality. So maybe those that fell below the red threshold before. And also sometimes if you get shorter read after trimming the adapters, you can also dump completely those sequences. Some research indicates that it is better to do gentle trimming, because before there was a very strict trimming based on quality, and it would remove a big part of your data. Yeah, we're going to use a skewer. Skewer is pretty fast and works well. When mapping to a reference, there are some things that must be taken into consideration, that we're working with RNA-seq data. That means it is RNA. If we're mapping to a genome, the genome contains full sequences of exons, introns, and our RNA comes from an actually spliced transcript. So if we were mapping the RNA to a transcript, there was no problem. It should map from beginning to end without gaps. But if we're mapping to a genome, we will have gaps. So we need to use a software that allows for spliced alignments. Also, if we were mapping DNA, it would be the same. We wouldn't have any problem of mapping to the genome. But it is not the case. Many times, if we have an annotated reference genome, we can provide the annotation so the software will know where or gaps, or introns and exons start and end to account for these intron-exon boundaries. But if we don't, there are also cufflinks, another program that, based on how your reads are mapped, it can try to generate some annotation file for you. And then the step that I was also saying, visualization. It is not usually a lot known, but it is very useful for you to know how are your reads distributed throughout the gene model that you provide. And also you can see if there is the splice sites are like you expected, or maybe you may have some isoforms, so you will see some mappings on regions where they were supposed not to. Sometimes it might happen that you have coverage only on one part of the genes, not all of that. This is mostly for visual inspection of your results after mapping. And finally, we're going to see differential expression. Many times you will have samples that you're going to have under different treatments or conditions, and you want to know what genes are being expressed differently from one to another. So in this case, like Meg was saying before, replicates will improve a lot more your results. Why? Because if you have few replicates, you won't really know if the differences are due to natural biological random variation, or also sometimes during library prep or sequencing, there are some slight differences that are random noise, and adding more replicates can help remove that. Here, for example, if I didn't tell you anything about sample 1 and sample 2, you could think two things, that they might either be biological variation, under same condition you can have different gene levels, or if they were two different treatments, they could actually be differential expression on mostly gene A. Also, one important thing about RNA-seq data, mostly compared with microarray data or some other sort for measuring differential expression, is that it is digital data. That means you're counting reads. So you are getting 1, 2, 3 x thousands of reads. You don't have a continuous variable. So you need some specific software that is able to deal with this kind of data. If you want to publish your results, like I said, you need to make your data public. As SRA, for Short Read Archive, can store two different types of data. First, your raw reads, and second, your alignments. You can store reads from many different sequencers, those that are here, 454, IronTorrent, Illumina. And then you need a lot of information before uploading to SRA. So it is better that you go to the NCBI website and find out what you need before, if you can, before starting your experiment, so you are sure to collect all the data that you need and also keep it updated. Many times it is a requirement before you can publish. And also, making your data public helps other researchers that may not have access to sequencing their own data. They use this public data to have their research done. And also, you might be looking at some characteristic, but some other researchers might be interested on the same data for analyzing something different. So it is very recommended. This is a general overview of how SRA looks like. You have mostly 4 tiers. first your bioproject, biosample, experiment, and runs. On your bioprojects, you have to describe what are the goals of your research. Be as more accurate as possible. Then you're going to describe your samples. They accept multiple kinds of organisms, also metadata of, whoops, if I see, yeah, like environment, yeah, metagenomes. And there you're going to describe your cultivars, your strains, if they were grown under some conditions of temperature, also if you have cell lines. So you have to be as much descriptive as you can with your samples. Then you have at the experiment level where you're going to indicate how the libraries were prepared, what are the sequencing protocols, and everything related to the generation of the actual data. And finally, you can have more than one run, but they all have to be uploaded to the same experiment. And yeah, there are multiple steps that they're trying to make it as much easier as they can, not for you to upload the data. Yeah. So the second part will be about trying to choose the best software for your analysis, because like I said, there are multiple software options at many of the steps on an RNA-seq pipeline. From our basic steps, there are, for example, for trimming, you can choose between Trimmomatics, Cure. For mapping, these are some of the most used software, like Star, Snap, Stampy, Top Hat. Maybe you have heard of SAM, maybe not. For differential expression, RNA-seq data, like I have said, it is digital data. So the negative binomial distribution is designed for deal specifically with this kind of data, and the most used packages are DSEQ2 and HR. But there are some others that you may choose. There are NoiseSeq, BaseSeq, SAMSeq, BMAC-AFDIF. And also for de novo assembly, there are multiple options. We're going to use Trinity, but for you to know that there are others like SOAP or TransAbyss. So, but in the end, there is like no best choice. They are all different. Some might suit better or worse your data. You have to compare. And there are some cases, some research that have used simulated data to try to compare their performances. For example, mapping. When you're mapping reads, you are trying to look at different parameters, like how accurate is the mapping, how fast is the software, how many resources does it consume, and yeah, so for measuring this, they have set different parameters, mostly from all your mapped reads, how many were correctly mapped for the precision. From your whole set of genes of reads, how many were also correctly mapped, that is recall. And also in the right figure, they have seen that errors and index make a big impact. On the left, this is from the STAR paper, where they were measuring how the ratio between the true and false positive rate when adding a different number of reads to the intronexon boundaries. They were trying to indicate that STAR, which is this one, was the best because it has the lowest false positive rate while maintaining the highest true positive rate. And also here you can see that different software behave differently when you are trying to add errors and index, and usually the average score is much lower in those cases. So depending on your data, you have to take these things into account. If you have a very close reference, you might not be very concerned about this, but if you are mapping into a more distant reference and you're expecting more SNPs, it is a big issue. Also, one might wonder, is it better to map to a genome or to a transcriptome? Because we have said that we can choose those both pathways. Yeah, well, it is not the same. It is interesting because they were comparing different software, and if you're only looking at mapping rate, in this example, Bauti were mapping more reads to the genome than to the transcriptome. So it is, yeah, to the transcriptome is mapping higher. I think I said it reverse. And to the genome, the mapping rate is a little bit less. While Stampy were mapping less reads to the transcriptome and more to the genome. And also, after doing the differential expression and comparing results, the vast majority of genes remain constant. But there are slight differences. Either you choose a mapping software or you choose mapping to the transcriptome or to the genome. So it is good to know what you're looking at and what results are the most accurate or you feel more confident with when you're doing your analysis. For differential expression, like I was saying before, having more replicates improves a lot the detection of differentially expressed genes. What you want to find when you're doing this analysis is to find the highest number of truly differentially expressed genes while reducing the amount of false positive differentially expressed genes. On the left, the figure is only indicating an overall score that combines both. They are separated with 2, 5, or 10 replicates. So as you increase the number of replicates, this relationship that about true and false positive genes rate is increased. And also here, they have separated the true and false positive rates to the right and the left. And when you increase the number of replicates, you will be detecting more. But many times, like it is not cheap many times to sequence, so you have to balance. In this paper, they were saying that at least you should have 6 replicates to improve. Also, if you are going to find genes that have a higher fold change, it will be more likely that they will be captured even though you have less replicates. But for those genes that are not varying much, you need more replicates to increase the power. Finally, for the novel assembly and annotations, there are again different things that you're going to look at. For your assembly, you want your reads to be as long as you can. Also, to have a good amount of transcripts. Trying to reduce the redundancy and that they're capturing the highest amount of reads. On this paper, the highest score was on both cases got with Trinity. So it is a good software choice. The Trinity score, it is a way of scoring transcriptomes. And for annotation, because like I said, if you have de novo sequence at de novo assembly, at first you don't know what those sequences are for. So you have to annotate. Usually you do some homology-based annotation. You would like that they contain an open reading frame, that they are the longest as possible, and also that they capture full sequences of homolog genes. And for the whole set of your transcriptome, there are some files that some search for essential biological functions. So you want it to be as complete as possible. They were comparing different approaches of combination of de novo assemblers with different annotation pipelines. Like you see, there is a lot of overlap. They were trying to indicate that this new methodology was the best. And it seems so. We have to try. So to conclude this part, there are multiple steps on an RNA-seq pipeline, and there are a lot of different options, and some are getting outdated, some are getting improved. So you have to see, it is good to know what works best for your data. In some cases, you can do some optimization, changing some parameters. Yeah, also you have to decide if you're going to use a genome or a transcriptome. Then for your differential expression, what algorithm you would like to choose. Like you have seen, your results won't be the same depending on your choices. So when you're deciding something, you can give some robustness to your decisions by, for example, making some tests on your data, and also looking at those research papers that are comparing software and say, okay, so I think this one might work the best, even though you're not going to try because you cannot try all the options available. So if you have any questions, if not, we're going to go through the first lab.
SRA templates. If you're about to start sequencing and you need help finding SRA templates and filling them out, let me know. I would much rather you figure that out ahead of time while you're doing your sequencing and come to me like 4 months later and ask for help submitting with SRA and like none of the information has been reported. That happens to me at least 2 or 3 times a year. It makes me very frustrated. So. Okay, so if anyone wants to run to the bathroom real quick or take like a minute or two, I'm gonna get started pretty fast since we're we've only got about 40 minutes left in the class, but I'll give you a minute or two. Okay, hold on. You guys have any questions about software choices? Is there anything you've seen that you wonder, like, what do you think about this software package? Or I have a particular problem? If your mapping really, if your reference is very distant from your the organism you're sequencing, like Miriam works on blueberries where she sequences different species, we use different breed mappers.
We're not going to cover these partially well. The methodology behind the methods. Because you can use a software package if you don't understand what it does.
I really recommend that you read the paper and read the manual. Read both. And the only way to really like dig into the underlying underpinning of the software. I talk a little bit about the underlying statistics, might be easy too, but it is pretty complicated. And I do it not justice. So it's worth reading the paper. The read numbers, it's good to understand how they work, but they're too standardized at this point. Like you're not the most you really need to understand about that is how they're handling multi-map reads or some of those ins and outs. But I mean, you could do some serious research in the algorithm, but they're so efficient. A lot of people are. Some people are working on that. Not too many.
Well, just to know the limitations, I think.
Yeah. I agree. I guess. Yeah.
Because, I mean, to what degree are we putting something in and getting something out?
Right, it's a black
There's
box.
always,
Really
but it's a black box of multiple levels, right?
Right, absolutely.
Until you give me a sequence of it, I say, I'm going to do everything myself. I don't believe I'm going to do everything in R.
Right.
I'm going to map things myself or something.
Right. You would be.
I would think about what statistic I should use, whatever this package is using.
Yeah. So yeah,
that's hardcore.
Reading the papers is, well, we usually dig into why they do
It's
what
not
they do.
enough. It's like the same thing. Read textbook and says every answer. I mean, you can't read. It's not enough. You have to do it. You have to do it. Test. You have to compare things. It's not enough.
Right.
Paper has graph.
Right.
Okay. What are the graphs? I don't know.
Yeah.
I mean, I can believe it. It works.
And we do that a little bit in my whole class, like take an actual read and manually map it back to Genome to see how that works. But it's yeah, it wastes like a whole class period. So all right, so let's log into Beacon. So have your keychain ready. I say as I like totally don't have my keychain ready. You can see my copy and paste. Okay, let's try that again. All right, and then I am going to use our new command, interactive, use our course allocation. We don't need 2.5 hours. We're only gonna be here for like another 40 minutes. So I'm gonna change that. Let's see if that works. Do I have to put the 00, Bobby? Yeah. I've never requested less than an hour.
You don't have to use it for 40.
Okay. And the shared access is important. All right. So if you're ever curious whether or not you're on the login node, your command prompt will actually tell you if this is login. You probably need to jump onto an interactive session. If it says something else, you know you're on a computational node, which is good. So as Bobby was telling us yesterday, we have some scratch space in Lustre Medusa, which is a good place for us to put our course files. And it will eventually get blasted away, so don't log in a year and expect that to still be there. Well, you won't have your student pins tokens anyway. But yeah, so this will all be practice. But if you ever want to get a copy of the files for your own use, just let me know. We're going to cd to Lustre Medusa, and I'm going to put mstate1 because that's where my directory is. You are going to put your student username, and if there's stuff in there, you can go ahead and wipe it out. So I'm going to remove -r RNAseq, sort of my scratch. You might want to remove -r star. I'll just get rid of everything. So now you should have a totally clean working directory. Everything you're deleting is probably from a student in a prior class. We assume that no, they don't need that data, it was scratch space, so do not feel bad just wiping it out. If anybody's having problems with this, or your directory doesn't seem to exist, your Lester Medusa student directory, red sticky, and somebody will come help you.
We just make the directory, or? I don't have.
Oh, I was just deleting everything. Like if you have stuff in there, get rid of it. If you ls and there's nothing there, you're good. Perfect. No, no, no. You should Lester, Medusa, and your student ID. This should exist. No. Okay. Will you help him copy? Okay, so I'm going to start by making a new directory, RNASeq, and then I'm going to cd into it. Now, inside of this directory, so this is, I like to keep my files really organized. So I'd like to keep my raw data in one folder. I like to keep each different analysis step in a different folder. This is going to help you a ton downstream. When you go back and you're trying to write the methods for your paper, you're trying to figure out what you did, what order you did it in. If you follow the example I'm going to give you, it's going to be much simpler. So I'm going to make another directory called raw data, and I'm going to cd into that. If one of the TAs wants to grab a marker and like draw the files as we create them, that might be helpful. So we have gone ahead and put some of Giuli's data into a global location that everybody should be able to copy. So we're going to copy Lester, Medusa, Proj, UT, raw data, star.tiny.fastq. And I'm going to copy it into my current directory. So tab complete is your friend on typing these nice long commands, so don't forget about that. And now if you do an ls, you should see something called earlyflower.rep1 and lateflower.rep1. Everyone was able to copy? Winjing, were you able to copy? Okay, perfect. She was having permissions issues yesterday, so we got those solved. Awesome. So this would be an example of the type of files that you would expect to get from your sequencing facility. Usually they're named in some odd way. Hopefully there's enough information for you to figure out what sample they correspond to. But the first thing I like to do is make sure these are actually FASTQ files, because you never know. So if I do a head, that's going to give me the first 10 lines. And indeed, I see our comment line, our first sequence, our completely useless plus line, and our quality values. And you'll remember that I like to see quality values that are capital letters. That means that we've got some pretty high quality bases going on. So just from looking at a single read, this looks pretty good. And then of course, because we got 10 lines, we have our second sequence, and then the third one. Now we're using a single-ended data set, so it's only one file. If you have paired in reads, you're gonna get two files. And so the first sequence in the first file, it's usually called like read 1 and read 2 or forward and reverse. The first sequence in the first file and the first sequence in the second file are paired ends. The next two are paired ends. The next two are paired ends. So they've separated your forward reads and your reverse reads into two different files, but they'll be in the same order so that you can reassociate them back together. And most software programs are going to expect that format, so you're not going to have to worry about it, but at least you'll know what you're looking at. Okay, and we can also do that for our late flowering and see that, yes, indeed, our late flowering is also, looks like a FASTQ file, so that's good news. Another tip, if you do a tail, if you do not have a complete sequence at the end, like if you have half of a line or not enough bases or not enough quality values, that can mean that your transfer died in the middle, and that's a bad sign. So doing a tail is always nice too, to make sure it looks like a complete file. So you should have a complete 4-line sequence at the end of the file. So we can look at how big our files are. You'll notice from our examples yesterday, when these numbers were super tiny, now we have much bigger numbers. And if we want to get that in a human readable format, we can do -LH. And that's going to tell you that each of these files is 39 megs. These are very, very tiny in the scheme of sequence files. We often get files that are many, many gigs each. So the LHS-LH will give you an idea of how big they are and how much storage space you need to be emailing NICs for to ask for. And another, so we know that there are 4 lines per read, so we can easily count how many reads we have as well. The command WC is the word count command, and if we give it the -L flag, it is actually going to count lines. So WC-L means count the lines in this file. And actually, and I'll show you how to use the star command as well. If I do star fastq, it's going to say for any file that ends in fastq, count the number of lines. And we have given you 1,000,000 lines in early flower and 1,000,000 lines in late flower. So how many reads would that be? 250,000. You have to divide by 40 at the number of reads because the reads, each read takes up 4 lines. All right, so let's start by doing some quality control. Oh, another way to count, oh no, that's, yeah. You can do the math on the command line, but I think it's easier to just divide by 4. Okay, so we are currently in raw data. Let's back up, back into RNA-seq. And now we're gonna create an analysis folder. And we're gonna CD into that. So one of the reasons I like to keep the raw data in its own folder, sequestered away from your analysis folder, is I think it makes you less likely to delete it or overwrite it. We won't talk too much about permissions in this class, but you can also set the permissions such that you yourself cannot overwrite that data without changing the permissions. And that can be a nice, if you're worried about doing that, that can be a nice fail-safe. Also, it's never a bad idea to keep a copy of your raw data somewhere else, like a hard drive in your lab. Okay, so we have our analysis folder, and then I'm going to make a folder called O1 FastQC. So you'll see as I think Fong and Sheely are drawing our directory structure, I'm going to make a different folder for each analysis that we do. Like I said, I just like to keep things super organized, and it's really going to make it easier on you. If you send these folders or these files to someone else, they're going to be able to follow along with what you did, what order you did it in. It's going to be very helpful. It's also a good idea, just because you're at a computer doesn't mean you shouldn't treat it like science. When you're in your lab, you probably keep a lab notebook. You need to write down what you're doing. You need to do that when you're doing your informatics analysis as well. So whether it's a Word document or a GitHub account or a text document, you need to be writing down what you're doing, why you're doing it, anything that went wrong, just so that you have a record. Okay, so we are going to run FastQC on each file. I'm going to use the full path to our install. Okay, so the only directory that doesn't tab complete for me is Proj. I don't know why, if you guys have the same problem, but the rest should tab complete. And this is why we really stressed that you needed to know absolute paths and relative paths. So this is our first complicated command we're seeing. I'm giving you an absolute path to some software that we installed in our course directory. We have also sent this to Bobby, and this may eventually be available through the module system, which we're not going to teach right now because we don't use anything currently in the module system. But it should be here for the time being if you want to use it for your own data. You can use this absolute path. And then we're also going to reference the location of our raw data, which is up 2 directories and down into the raw data directory. Specify directory. Oh, I have a -O in there. Take the -O out. Oh, how are we going to do that? Okay. So if you get rid of the raw data and we do early flowering. Let's see how that works. This should run the software. Okay, so this ran on early flowering. It's going to give you a % complete. This went really fast because it was very small. If you have a large file, this can take 15, 20, 30 minutes. So be patient, request nice long sessions, and eventually it will return you to your command line prompt. Okay, and now what happened, this command was completely formatted wrong. I'm sorry, guys. If we actually look in the raw data folder, it created our fastqc.zip and our fastqc.html. So that -o, that flag that I originally tried, I thought it was going to move the files in this directory, but it didn't. I'm going to have to go back and read the manual and figure out how to actually do that.
Actually, put down the website, right? You forgot, I think, -o, for the -o, just a period.
Oh, a single dot. Good job, guys. Okay, so, but again, this is why it's really convenient to be able to navigate the file system when you realize you've screwed up. I forgot that extra 1 dot in the middle. That's what I did. I didn't even see it when I was looking at the notes. But I can just move those files in here. Rep1.tiny. And here they are. So if you accidentally create a file somewhere that you don't want it, you can just move it over to where you do want it.
Yeah. Oh,
and we can run our other one. So now that we've realized my mistake, I forgot that dot. We've already run early, so now we can run late. So I'm going to specify the software. I'm going to say put the output in this directory. That's what that dot is, except it was hard to see because it's right mushed up against, and then go find the file that's back to directories in the raw data folder called late file. And now if we ls, we see that we've created an HTML file and a zip file. So while I love fastqc, it is super weird that it is creating output in HTML. That's just what it does. Everybody loves it. But HTML is not something you can really visualize on the command line. So at this point, if you only had 2 files, I would transfer them to my laptop and look at them. But the reality is oftentimes you have 100 files and you might run this 100 times, and then we're going to show you how to basically aggregate all of that data into one file. Right now it's just gathering statistics. It's not changing your input, it's not doing anything, it's just gathering statistics. Oh, right, so, okay, so let me see if I can show you, and this will become more clear when we look at the output. So Babraham Bioinformatics are the people who produce this. And actually, it's really nice because they have an example of good Illumina data and an example of bad Illumina data. So let's look at the bad example because that's always more fun. So they give you some basic like total number of sequences, sequence length, %GC. They show you the Venn diagram of your sequence quality. So as the read is getting longer, you're getting a lot more variability in the quality. You still have your average quality is dropping. You still have some good quality bases up here in the green, but you have a lot of bad quality bases as well. This is what I was telling you about the glass slide. So when the slide goes on the instrument, it can tell you like this region of the slide seems to have some problems, this region of the slide. And if you see like the whole slide that's half red, then you can usually like call the sequencing facility and be like, something went vastly wrong with your sequencing. And you should give me a free run. Sometimes they buy that. It'll also give you per sequence quality score, so a little bit more information about average quality per read. Your GC content, which usually should be pretty standard across the whole read. Sometimes you'll get extra variation right here because the random primer ligation it's not actually truly random. So sometimes you do get some weird GC stuff in like your first 6 spaces, but the rest of it should be pretty stable. Your GC content. So again, this might rely, if you thought this may or may not be normal, you might need to know a little bit about your organism. Do you have a skewed GC distribution, or is it pretty normal? Any ends, this will usually almost never show up in a Illumina data set, any uncalled bases. The length of the sequence, duplication levels, and RNA-seq, you're gonna see a lot of duplicated sequences. And also your overrepresented sequences, oftentimes these are adapters. So you can figure out if you have anything that's weirdly overrepresented. The software will also check for some of the primary, most common adapters, tell you if you have any, and do a k-mer analysis. So look for overrepresented k-mers. Usually if you see something like this, it's some kind of adapter weirdness, where you've got an adapter that hasn't been appropriately clipped out. So right now we're running fast QC. We have not done anything else. We haven't trimmed adapters. We haven't done any other QC. We're just gathering all of this information. Yeah, so we're gonna skip transferring to our own computer because we're gonna do that at the end of the lesson, and I'm gonna let Bong take over and go through how to do trimming. So it would be a good idea at this point, you may not need to trim, so you might wanna look at your fast QC before you trim, but we're gonna show you trimming and then show you how to aggregate all of the information and
You have the instructions which install that.
install instructions? Yes, I can share those with you. Fast QC in particular is super easy. You just download it, stick it in a directory somewhere, and it just works. Not all of them are that easy, unfortunately, but Fast QC is pretty easy.
Full command line.
Yeah, it depends on the software. Some of them
Yeah,
will
fast
need. Oh, you do? You have to do extra stuff.
Just a little.
So when I start working with sequencing data, I always kind of keep a concept in mind. So it's garbage in will be garbage out. So before we do the real analysis with the sequence data, we want to make sure that the sequence are cleaned up, the low quality sequences are removed. So to achieve this goal, we're going to use a skewer to do the trimming. So now, if you press PWD to check for your, so now you may be inside of the 00 fast QC folder. So we want to change our folder to analysis. Go up and make sure you are inside of the analysis folder. And now we make another folder inside of analysis. It's O2 skewer. And we want to change directory to this folder. So now we can run the skewer with our raw data, which is stored in the 00 raw, which is stored in the raw data directory. So just copy that. Oh, no. So I'm going to copy this whole bunch of command into my command line. So enter. Now it's running the skewer. So so this process may take several minutes. So during this process, I'll give a explanation of each of these parameters we use. So the first line give a path of the software of skewer. The second parameter -L specifies the minimum length of the sequences we want to keep after trimming. Here we set it to 30 base pair. The default value is 18. So the -O flag means it will specify the prefix of the output file. And the -X means you specifies what are the adapter sequences you will give skewer to trim off. So here we point it to adapter FASTA file. And then this command line gives the raw data. What is the FASTQ file you want to trim off? So it points to the raw data file and the early flower FASTQ. And here this more than symbol means we're gonna to store our output into this early flower.out this file instead of output them on the screen. So now let's go back and have a look. Now it's already done. If we ls, you will see it has output file, the trimmed FASTQ file, and the log file. And the same thing with the late flower file. I'm gonna copy this and run the same thing on the command line. Enter. So you may wonder what are all those /means. So in fact, we can write this whole bunch of commands just in one single giant line, but it is not very easy to understand and to read this. So we give it like a separator, to the command to tell the command that my typing hasn't finished. So you need to wait until I finish the whole command line typing. Does that make sense? Okay, it's already finished. So now let's look at what are the output inside of the .out file. So we stored our output in this file. So we'll use nano.out. So if we use nano, it will give the whole bunch of the files. So we don't want to go through all of this, use the arrows. So I'm going to use tail and just look at the last part of this file. And you can see here is a brief summary of the reads after trimming. So the total reads are 250,000, and only one short read is filled out after trimming. If you remember, we set the minimum length that will be kept in the trimmed file is 30. So maybe after trimming, there is this one read is shorter than 30. So this is removed. And about 20% of the reads are trimmed, and 80% of the reads are kept without trimming. And also, you can go inside of the log file and have a look what is the parameter setup you used. Okay. Oh, okay. Yeah. Inside of this log file, you will see all of your parameters you set out for this skewer process. So these are all the sequences of the adapter sequences. And then you can see the minimum error ratio you allowed it for 0.1 and the minimum interval error, the read length. And at the end, it will give the summary of the trimmed sequences. And at the very end, it will give the length distribution after trimming. So as you can see, the minimum length of trimming is 32, and the maximum is 51. And most of the reads, even after trimming, is around like 47 to 51 is.
20% normal for the amount that gets trimmed.
You mean,
Actually, I think it's kind of good. Normally we trim a bit more. So it just depends on how good the sequence you want it. So not all of them have adapters. Like sometimes the adapter doesn't make it. Sometimes the sequencing facility is not for you. So usually we look and see those past QC files, which we haven't looked at. But I normally look at those first. But if you look at these, there are no adapters. So it's mainly trimming quality, anything that's low quality. Yeah. No, but most. But occasionally they won't. It's always worth checking, trust me.
yeah, usually after the sequencing, they will trim the adapters for you.
It's usual, but and skewer, it's not going to hurt you to check, right? Like you might as well just check. Sometimes it can be difficult to get the adapter sequences from the sequencing facility. So sometimes you just have to figure out what kit they use and then Google around and see if you can find your adapter sequences. But yeah, worth doing.
Yeah, so in our case, it just trimmed the low quality sequences. So now we are inside of the O2 skewer folder. We want to do a multi-QC analysis. So we're gonna to get out of this folder and change it to the analysis folder. Now we want to make another folder named O3 multi-QC. And change it right to this one. So the advantage of multi-QC is it can aggregate the multiple bioinformatic results into a single report. It is pretty neat. So if you, it is supported several tools. Here you can see it can support tools include this bunch of softwares. So it will aggregate the information of all of those softwares and generate a single report to you. So here, so first we need to link the output files from the FastQC and the skewer to the current directory. So we're gonna use ln, it means link, and -s means soft link. So we're going to link these files into the current current directory. And now if you ls, you will see this linked file is already here. But if you want to see what is the actual path of this FastQC, you can use ls-hl. You can see this file is actually sit inside of this folder. Although you can use this file, but actually it is existed in the in this directory. We will do the same thing to the skewer folder. So else. And now we will link the skewer files into the current directory. And now if you else, you will see the O2 skewer directory. Same thing, you can check the original location of this folder. And now we are ready to run the multi-QC software.
I think you were supposed to link the files on the number.
Yeah, she missed the start of the 01-FastQC.
Oh.
You create the name.
Yes. So you are supposed to link all of the files inside of this folder to the current directory. Otherwise, it will not read the files. So I'm going to.
If that, you can just use RA to remove those two, the subdomains, and then we do that.
Okay, now we will remove all of these things inside of this folder. Now it's empty, so I will link this again. ln-s. Yeah, here we need to add a star, means we want to link all of the files inside of this folder to the current directory. Now it should be like this. And the same thing to all of the files inside of a skewer file. Now you can see all of these FastQ files, the HTML, the FastQ output, and the traumatic output are inside of this folder. So multi-QC will use these results, generate a single report. So now we are going to run the multi-QC. Now it's running. It will take some minutes to finish this.
Great, so we're essentially out of time. So if you have time to let this run and complete, that's great. And the homework for tonight was to send me your multi-QC file, which we won't get to FileZilla today, or if you secure copy preferred, way to transfer files. So we'll do that tomorrow morning, and then you can just email me your files. So don't worry about emailing your multi-QC file unless you're just super savvy and know how to move them around. But once it completes, I think you guys are good to go. But hang out if you have any questions or want more clarifications. I've had a number of people asking more details about how the software works. So you can definitely go through that. And, yep, thank you.
What year?